In this presentation, we'll look at how cells convert chemical energy from glucose into ATP. Cellular respiration is the process by which cells convert energy stored in chemical bonds into ATP. Remember that we can think of ATP as being the energy currency of the cell. So by converting sugars into ATP, that energy becomes available for many different cellular processes. This is a pretty complicated process, so I wanted to just start off with a very brief introduction so that as we go through the details, you don't get lost in them. So we start this process with a molecule of glucose that has been transported into the cell. A process called glycolysis breaks up this glucose molecule and in the process generates a little bit of ATP and transfers electrons onto a molecule called NADH. You can think of this NADH as a high-energy electron carrier. The result of breaking up the glucose molecule is pyruvate, which is then transferred into the mitochondria of the cell, where it's processed into acetyl-CoA. This acetyl-CoA, or coenzyme A, then goes through a process of breaking down into carbon dioxide molecules in the citric acid cycle. The citric acid cycle produces more ATP, but mostly it transfers electrons onto NADH and a similar molecule called FADH2. These are both high-energy electron carriers. These molecules carry their high-energy electrons to the inner membrane of the mitochondria, where they enter the electron transfer chain. The final result of the electron transport chain is the production of lots of ATP through a process called oxidative phosphorylation. We'll take each of these steps in turn, but first we need to learn a bit about redox reactions, reactions which involve a transfer of electrons. One important way in which molecules transfer energy is by transferring electrons during chemical reactions. Reactions like this are called oxidation-reduction reactions, or redox reactions for short. In this simple example, the sodium atom loses an electron and the chlorine atom gains an electron. We say that the sodium is oxidized and the chlorine is reduced. To help you remember which is which, you can remember oil rig, where oil stands for oxidation is loss, and rig, reduction, is gain. And here we're talking about electrons, of course. You can also think of reduction as reducing the charge, that is, making it more negative. Redox reactions don't need to involve a complete transfer of electrons. Reactions in which electrons are moved further away from an atom or closer to an atom are also considered redox reactions. In this example here, methane has a carbon atom that shares electrons with hydrogen atoms. Both atoms have approximately the same electronegativity and therefore share electrons approximately equally. When methane reacts with oxygen, the result is carbon dioxide. The carbon-oxygen bonds in carbon dioxide are very polar, with oxygen attracting the electrons much more strongly than carbon. Therefore, we can say that the carbon in methane has been oxidized. That is, it has lost electrons as those electrons move further away from the carbon atom towards the oxygen atoms. Similarly, the oxygen molecule, O2, shares electrons equally between the two oxygen atoms. The oxygen molecule gets reduced to water, in which the oxygen attracts the electrons much more strongly than the hydrogen atoms. This gain of electrons is reduction. We can see the same thing happening in the oxidation of sugar molecules. If we were to attract the electrons in the same way, we would see that the glucose molecule here is oxidized to carbon dioxide, and the oxygen molecule is reduced to water. Now, although this isn't a hard and fast rule because the definition of redox only involves a transfer of electrons, you can often think of redox in terms of whether oxygen or hydrogen atoms are involved. The glucose here gets more oxygen atoms attached per carbon atom, and it is oxidized. The oxygen molecule gets hydrogens attached, and it is reduced. As we'll see in a minute, the addition of electrons to atoms as a molecule gets reduced may or may not be accompanied by a transfer of the hydrogen nucleus itself. Let's look at redox reactions in a context of cellular respiration. When you eat food, electrons are taken from the food and transferred onto a chemical called nicotinamide adenine dinucleotide, or NAD+ for short. 2 electrons and 1 hydrogen nucleus are added to the NAD+ to form NADH, and the other hydrogen ion is added to the aqueous surroundings. You can see here where that positive charge comes from. That it comes from the nitrogen atom in the NAD+. And when the molecule has accepted 2 electrons and 1 proton, it has an additional hydrogen, and the nitrogen is no longer positive. So NAD+ can be reduced into NADH very easily. And the reverse is also true. NADH can be oxidized into NAD+. This ability to accept and release electrons makes this a very useful electron carrier, as we'll see later. In a similar way, FAD is another molecule that can accept electrons to be reduced to FADH2 and then oxidized back to FAD. One of the most important things you can remember from this is that the reduced form of a molecule, the form that's carrying electrons, is the form with high energy. So let's look at how NAD+ gets reduced, that is, how it gets its energy. As we've already seen, as we eat food, we metabolize that food and get electrons from that food. This is another case of coupled reactions, similar to what we have seen before with ATP. The electrons in the high energy food are transferred to NAD+ to reduce it to NADH. So remember, NADH, the reduced form, is the high energy form. And this will make sense when we look at electron transfer in the electron transport chain. As we walk our way through cellular respiration, remember that both NADH and FADH2 are both the reduced high energy electron carriers. FAD and NAD+ are the oxidized low energy forms. The first steps of breaking down glucose is a series of reactions collectively called glycolysis. So what we're going to be talking about now are these reactions right here. Okay, before you look at this and panic, let me just put you at ease and say that for my classes, you don't need to memorize all of these reactions. I will give you hints along the way as to what you need to know by heart and what you don't. Glycolysis occurs in the cytosol of the cell. We'll move to the mitochondria later, but for glycolysis, it's all happening in the cytosol. It's convenient to think of glycolysis as consisting of 2 phases. The first of these phases is called the energy investment phase, and it's called this because we actually need to input energy in order to break up the glucose. Remember that glucose is quite a stable molecule, and so to be broken up, it needs a little bit of a push. That push comes in the form of ATP and phosphate groups being added onto the glucose molecule. In 2 different steps, phosphate groups are added to the glucose molecule until we get fructose 1,6-bisphosphate. Notice that each of these reactions is an enzymatically catalyzed reaction. Every single one needs a special enzyme in order to carry out that reaction at an appreciable rate. Also, notice that reactions that involve ATP, or the transfer of a phosphate group, are all catalyzed by enzymes called kinases. You should know that kinases facilitate the transfer of phosphate groups. Glycolysis starts with glucose, a 6-carbon sugar. Ultimately, at the end of the energy investment phase, glucose is split into two 3-carbon molecules, DHAP and glyceraldehyde 3-phosphate. Keep your eye on glyceraldehyde 3-phosphate, G3P. We'll see it again later, including in photosynthesis. Notice that in this picture, there seems to be a reversible reaction between the DHAP and the G3P. These two molecules are easily converted one to the other, but it's only G3P that's used later on in the next steps. Because of this reversible reaction, DHAP that is produced can easily get transformed into G3P, and G3P gets used up later. So in summary, the energy investment phase involves phosphorylation of glucose, rearranging, and finally splitting the molecule into two molecules of G3P. The next phase is called the energy payoff phase, and in this phase, G3P is rearranged ultimately to pyruvate, and in the process, we gain a few molecules of ATP. We also gain a few molecules of the energetic electron carrier NADH. Let's do a little bit of accounting. For each molecule of glucose, there are 2 molecules of G3P produced in the energy investment phase. Each of those molecules enters the energy payoff phase, and each produces 1 molecule of NADH and 2 molecules of ATP. Therefore, a total of 2 molecules of NADH and 4 molecules of ATP are produced. Thus, overall, glycolysis produces 2 molecules of NADH and 2 molecules of ATP for every molecule of glucose. Finally, of course, it also produces 2 molecules of pyruvate, which is used in the next steps. Before we move on, let's look briefly at the process of transferring a phosphate group from one molecule to the other. As I mentioned earlier, this is carried out by an enzyme called a kinase, and it simply involves moving the phosphate group from one molecule to the other. Now, because the phosphate's originally attached to another molecule, which we call a substrate because it's part of an enzymatically controlled reaction, this is called substrate-level phosphorylation. Let's look at one of the reactions in the energy payoff phase that we just saw. In this case, the 1,3-bisphosphoglycerol is the substrate and has attached phosphate groups. The kinase binds the substrate and the ADP and then transfers the phosphate group to the ADP to make ATP. Okay, so just to track where we are in this long process, we've covered glycolysis, which produces ATP, NADH, and pyruvate. Remember, the glycolysis happens in the cytosol, and therefore the pyruvate is produced in the cytosol. To complete its oxidation, the pyruvate needs to be transferred into the mitochondria, and this happens via active transport, which requires a little bit of energy in the form of ATP. As we've already learned, the mitochondria is often referred to as the powerhouse of the cell because this is where most ATP is generated. The structure of the mitochondria is really important with regard to how it carries out those functions. The mitochondria has 2 membranes, an outer membrane and an inner membrane. The inner membrane is infolded and joined with an extensive membrane structure that goes throughout the mitochondria. These infolded membranes are called cristae, and they house many of the enzymes and proteins that we'll talk about soon. The space between the membranes, the intermembrane space, is also very important for reasons that will become clear in a few minutes. For now, we're more interested in the mitochondrial matrix, which is the liquid portion of the mitochondria that is fully inside both membranes. Thus, the pyruvate must be transported through both membranes and ends up in the matrix, where it will be oxidized in the next steps. So, the pyruvate enters the matrix of the mitochondria and in 3 steps is oxidized to acetyl-CoA. In the process, a molecule of carbon dioxide is lost, and the 3-carbon pyruvate is turned into a 2-carbon molecule attached to coenzyme A. A molecule of NADH is also formed, Again, remember that NADH is a high energy electron carrier. The resulting molecule is attached to coenzyme A to form acetyl-CoA. If you're counting molecules, remember that for each molecule of glucose, 2 pyruvate molecules are produced. Therefore, oxidation of pyruvate produces 2 carbon dioxides and 2 molecules of NADH and 2 molecules of acetyl-CoA. The carbon dioxide that's produced will eventually be breathed out. The acetyl-CoA molecules then enter the citric acid cycle, where they're broken down progressively into smaller and smaller molecules. As you can see here again, the citric acid cycle takes place in the matrix of the mitochondria and produces ATP and electron carriers into the matrix. Like with glycolysis, the citric acid cycle is very complex, and we don't need to worry about learning all of these reactions. What's important to learn is that as acetyl-CoA enters the citric acid cycle, it's attached to oxaloacetate to produce citrate. This is then progressively rearranged and broken down back into oxaloacetate. As this rearranging progresses, the carbon backbone gets shorter as carbon dioxide breaks off. Several of the steps involve changes in energy, and that energy is saved as either NADH, FADH2, or ATP. Acetyl-CoA is a 2-carbon molecule attached to coenzyme A. The coenzyme doesn't really play a role in the citric acid cycle, and it's returned. Therefore, at the beginning of the cycle, a 2-carbon molecule effectively joins with a 4-carbon molecule, oxaloacetate, to form the 6-carbon molecule citrate. As the cycle progresses, you can identify 2 reactions in which carbon dioxide is released. These 2 molecules of carbon dioxide account for the 2 carbon atoms that entered as acetyl-CoA, although technically they aren't exactly the same atoms. There's one little idiosyncrasy that you should be aware of. You can see in step 5 that ATP isn't produced directly. Rather, GTP is produced by phosphorylating GDP. GDP and GTP are completely analogous to ATP and ADP, except that the nitrogenous base is guanine instead of adenine. At any rate, GTP can go on to phosphorylate ADP into ATP. So let's continue our accounting. For each molecule of acetyl-CoA that enters the cycle, 2 molecules of carbon dioxide are produced, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of ATP. Remember, though, that we should double all of these numbers because for each molecule of glucose, 2 acetyl-CoA molecules are produced, and the cycle turns twice. Okay, that's about it for glycolysis, pyruvate oxidation, and the citric acid cycle. Just to recap, we started with a molecule of glucose and split that into 2 pyruvate molecules. These pyruvate molecules were shuttled into the matrix of the mitochondria and oxidized to form acetyl-CoA. The acetyl-CoA enters the citric acid cycle where it loses its last 2 carbon atoms and generates a bit of ATP, but many molecules of the reduced electron-carrying molecules FADH2 and NADH. Before we leave this, I want to mention that the energy investment phase of glycolysis involves endergonic, non-spontaneous reactions. However, every subsequent reaction is exergonic and therefore spontaneous. The free energy contained within the glucose molecule is gradually stripped away, but much of the energy is saved for use by the cell, either in the form of ATP or, more commonly, in the form of one of the 2 high-energy reduced electron carriers, FADH2 and NADH. In the next presentation, we'll look at what happens to those electrons in the electron transport chain and how chemiosmosis is used to produce ATP by oxidative phosphorylation.